Our next talk is from Madeleine Henry, who is from the Scottish Rural College in Inverness, and she's going to give us a talk on the prevalence of verocytotoxigenic E. coli O157 in British cattle populations.
Thank you. The chairman and the keynote speaker here today both lectured me at the vet school here in Dublin, so you have them to blame for this. Apologies in advance. I'm going to tell you a little bit about some work we've been doing at the Epidemiology Research Unit of SRUC, which is in Inverness in Scotland. And I'm going to talk about E. coli. We're all quite familiar with E. coli as practitioners. There are various cases where it's problematic in cows, but today I'm talking about E. coli O157H7, and particularly the strains that produce verocytotoxin, which we're dealing with as a human pathogen. It's relevant to cattle practitioners because it's in cattle, and they're recognised as a reservoir for human infection. Cattle, the organism can live in them apparently without any issue for the cattle. It's shed in the faeces, and contact either directly or indirectly with human feces or with cattle feces is how humans become infected. In adults and immunocompetent individuals, that can be quite a simple infection, just a bout of diarrhea, but in young children, the elderly, or people who are immune suppressed for another reason, it can be severe or even fatal. So it is quite an important public health issue. In Scotland, there have been 2 prevalence surveys done in cattle for that are being produced for human consumption. The acronyms that you see there just refer to the funding bodies that were involved, but if you do read any literature about these, you'll often find them referred to in that way. In both of these studies, it was possible to detect O157 on approximately 20% of the farms that were sampled. And the original CIRAD study involved just under 1,000 farms. The IPRAVE study involved just under 500 of the same farms that were revisited. Okay, there hasn't really been found any successful cattle level intervention yet that's kind of feasible on farm in dealing with O157. So the messages on how to control it are general public health messages, and the front one you see there is the Clean Livestock Policy in the UK. The human picture, which is recorded by DEFRA in their zoonosis report, has been broadly similar since 2004 in this graph, which is, as you'll remember, the end of the last survey in cattle in Scotland. You can see in the red line, which is the top line, that Scotland consistently has a higher rate per 100,000 population than the other countries in the UK. That spike in Northern Ireland related to a foodborne outbreak that happened a couple of years ago. There's been work done in the intervening period in human isolates of O157, and it's been shown that the organism is evolving. The types that are causing cases in humans are changing somewhat, but we have no picture of what's been happening in the cattle. So the aim of this work that I'm talking about was to conduct another prevalence survey in Scotland, going back to a subset of the farms that had been visited previously, and also to conduct a prevalence survey across England and Wales as one sampling unit, which would be comparable to the survey in Scotland. And we did this over the period of 1 year and 1 year and 3 months for England and Wales in between 2014 and 15. We sampled cattle on the farm, and our target group was the group that was closest to slaughter on the day of sampling. This was the group that had been targeted in the previous 2 Scottish surveys, so our aim was to make our methods comparable. The samples that we collected were fecal pat samples from the area where the cattle were kept. If they were indoors, it was the pen. If they were outdoors, it was the pasture. That didn't matter. The number of samples we collected was determined by the number in the group, and we had a 90% power of detecting O157 on the farm if at least one animal in the group were shedding. We went to 110 farms in Scotland, as I said, a subset of the original farms, and 160 farms in England and Wales. As well as collecting samples, we also conducted a questionnaire with the farmer or the animal management staff there, and this was to gather demographic and management data that were relevant to O157 status. Analysis-wise, back at the lab in Inverness, the samples were cultured, VTEC was identified if present and enumerated, And any positive isolates were also run through a PCR test to determine which type of verotoxin they produced. VT2 was the most common, also VT1, you might see those in the literature. We also did some statistical analyses. Essentially, we were aiming to come up with a prevalence estimate, and we wanted to take into account what we'd found from our farm management questionnaire in terms of demographic data and also seasonal sampling in our eventual prevalence estimate. We treated Scotland and England and Wales as two separate units because the way we'd gone about identifying the farms to sample was slightly different for the two. So we have two surveys. And our results, 1 in 5, approximately speaking, of farms in both Scotland and in England and Wales were found to be positive, where we're defining a positive farm as a farm on which at least one sample tested positive for O157. This result is broadly comparable with what we found in Scotland in the two previous surveys, and it's interesting that we've also found quite similar results in Scotland and in England and Wales. There have been surveys conducted in England and Wales for O157, but they haven't been quite exactly the same methods, and sometimes the results have been a little bit different. So it seems, anyway, from the face of it, that there hasn't been a drastic change in the picture on Scottish farms since the previous surveys about 10 or nearly 15 years ago. We've also had a brief look, although the work is still ongoing, at the proportion of samples that were collected overall that were positive, and it does appear that there's been a change there, but I don't have enough information to interpret that for you at the moment because it's really a function of not only the herd prevalence but the within herd prevalence. So the work that we're going to do next, we'll have to investigate that further. This project that we've been working on, the field study, is part of a consortium project that's funded by the Food Standards Agency and Food Standards Scotland. You can see there that we have a lot of consortium partners in this, and there are a lot of different aspects to this work. Our prevalence estimates and also the E. coli isolates and samples that we've collected are going to be used by colleagues at these other institutions, essentially to do a comparative analysis of what we're finding in Scotland, in England and Wales in cattle between those two units, what we're finding now in Scotland compared with what we found before, and also to compare human and cattle isolates. In the previous surveys in Scotland, the isolates from cattle were very similar to the isolates from hospitalised human cases at the time. As I said, we don't have interim information, but we know that the human picture has changed. So colleagues in this project will be doing whole genome sequencing and other techniques to determine whether the picture has also changed in the cattle. The ultimate goal of the trial, of the project, is to come up with some proposed interventions, to model them, find one that looks likely to be the most effective and feasible, and then trial that preferred method. So no doubt there'll be more information at a future time. That's me. Thank you for listening, and to all the farmers that participated. And I got to drive around Scotland and see views like this, which was probably the best part of my job for quite a while. Thank you very much for your attention. Cheers.
Well done, Madeleine. Thank you very much. Are there any questions for Madeleine? Yes.
Hi, Clarice Lulayanji from Canada. You mentioned the clean livestock policy in the UK. Do you have any sort of really general information on how it affects, like the effect that that policy has in general in terms of human cases of E. coli? Because it just seems like a very loose is not the right word, but it seems very naive to think that just because the cattle are clean that they are not carrying E. coli or 157.
No, yes, I see what you're saying. No, I think that is the point, really, that there hasn't been anything that's targeted enough for E. coli, particularly a bacteria that has such a low infective dose and which we have to assume to be everywhere, because sampling a farm one day and not finding it doesn't tell you that it's not there. So yes, absolutely, that is a good point. It's not really enough to just say, oh, make sure you do this, that and the other and you'll be okay. We do need to find some specifically targeted interventions, which is where the work is going in the end. Thank you.
Patrick Duggan, Tipperary, Ireland. I was just wondering there, and it's particularly because a child of a client picked up E. coli this year, And I know you mentioned that all the sampling was done at cattle prior to slaughter or within that 6-month period. Has any work been done on younger animals, or how young can, say for argument's sake, calves have picked this up within a herd? And particularly where you've got kids on family farms still, where they do go out and about, it still happens in a lot of places. To what extent would you consider that they might be at risk?
I would think you would have to assume they're always at risk. There has been work done which showed that weaning time was a particularly high risk period in terms of finding it in the cattle, and that it would taper off after that. But it's also been isolated from younger calves as well. And again, because of the low infective dose, etc., it's really not safe to assume that there's any age of animal that's a no risk age. So yes, you're quite right, and I think that's why sometimes vets have so many, and farmers have so many different bugs and pathogens to take into consideration, and this might not be the highest on their priority list, but it can be so disastrous if it does happen, if a child is affected, that they really just need to be aware of that, because what might happen could, just be catastrophic.
Mario Luini from Italy. Have you any data on E. coli, verocytotoxigenic E. coli, different from O157, such as O26 and other that actually in Italy, but also in Europe, are more relevant in human illness?
No, not personally. I don't know. I am aware of that, and other people in the group have worked on those other strains, but I won't pretend I can give you information on it because I can't. Sorry.
I'll just add to that, the samples that have been collected within this study are being used for non-O157 and other STEC studies under a Wellcome Trust grant by a postdoc there, so there will be further details on that.
That's why it's good to have colleagues here to help.
I might also comment, there's a survey in abattoirs that has just been completed here in Ireland looking at O157 and some of the other VTEC serotypes, and that material is currently being published. There are other serotypes being detected, but at a far lower prevalence than O157. Okay, Madeleine, thank you very much
Thank
indeed.
you. Cheers.